inviting me to present today. The topic of my conversation is neurobiology of SARS CoV-2. I have no conflicts of interest to declare. So this is our version of a coronavirus. This was done by a tattoo artist in July 2020 who was put out of business by the pandemic, and he felt as his contribution he would start making art about the pandemic. And I think this is a good depiction of what we think about the coronavirus 19 vaccine. So I've been interested in coronaviruses for a long time. And this image is from a mouse infected with a mouse coronavirus, not a human one. And it's from, probably 20, 25 years ago. And what it shows is really a summary of what I think coronaviruses do. So if you look in the middle panels here, which says RAG1 knockout, this mouse is infected with this murine coronavirus, but it has no T or B cells. So it can't really clear the virus. And the virus is all along the white matter of the spinal cord. And the white matter looks pretty good. And then if you add back T cells, within 7 days, the virus is gone, that's shown here, but now the myelin is completely destroyed. And the axons under the myelin are also damaged. So the question is, why can't this host clear this virus without causing much damage concomitantly? And this is something that I think is also a feature of COVID-19, host immune-mediated damage. So I wanted to go briefly over what we knew about coronavirus infections of the human CNS before SARS CoV-2. We know that OC43 was postulated to be a factor in initiation or exacerbation of multiple sclerosis. OC43 is a common cold coronavirus. However, this is not supported by data, but this is something that's been in the literature for a while. And there's one case of lethal OC43-induced encephalitis. So the bottom line of this is that there's very little evidence that the common cold coronaviruses actually cause infection of the brain or spinal cord. This is the one case that I just referred to. This is a baby who has no T or B cells, a baby with severe combined immunodeficiency. And this baby died from OC43-associated encephalitis. And what you can see here is evidence of virus staining throughout the brain. That's the brown cells here, shown in D. And the other infections that preceded SARS-CoV-2 included SARS. And we know after SARS, some patients developed acute psychosis. This was attributed to steroid use, personal vulnerability, likelihood of developing psychotic reactions to being in the ICU or being on steroids, and psychosocial stressors. And occasionally there were reports of viral RNA or protein in the brain on autopsy. However, I'll show that in the next slide or two, and this was shown really rarely, so I'm not sure it's true. And some SARS survivors had neurologic psychiatric complaints. And at the time, it was attributed to steroid use and ventilator use. This is the case that I was referring to. There's very little other cases, as I mentioned. And what you can see here in O is putative viral staining, or viral antigen staining in the brain. Whether this is real or not is a little hard to tell from the manuscript. And certainly since it wasn't found at all commonly, it's really questionable about whether the brain was involved in SARS or not. So this is another case from SARS. This is a 39-year-old physician who more convincingly has viral antigen in his brain after he succumbed to the infection. MERS, which is the other major human coronavirus infection, is pretty rare, occurs about 2 cases a week now, a total of 2,500 cases in the 9 years since it's been first described, but it has a very high mortality, 35%. Neurological disease is very rare. There were 3 patients reported with acute neurological disease, but it's also not very common. And again, survivors had neurological and psychiatric disease. And this may have been, again, attributed to ventilator use or steroid use. And only 2 autopsies were performed, so we really have no information on brains at the time of death. So in terms of COVID-19, as I think everyone knows, there's been many neurological and psychiatric sequelae recognized. These include entities such as brain fog, cognitive defects, encephalopathy, stroke, seizures, Guillain-Barré syndrome, anosmia, geusia, and maybe an increased risk of neurodegenerative disease, though it may be too early to know this. And the thing about the COVID-19 pandemic, certainly acutely, it's been a disaster, but long-term sequelae may be even more important because these may last for several months and be disabling. They may involve the brain, as I mentioned already, also the heart, the kidney, the lungs. The pathogenesis is not understood, and there's big initiatives now to try to understand what the basis is of long COVID or long hauler syndrome. But this will be a long-term effect. There are clinics in many places now trying to help patients who have this. And one thing that's striking in all these organs that are affected is that there's almost no evidence of virus infection. Rather, we attribute this to combinations of pro-inflammatory mediators, thrombosis, vasculitis. How this is working, though, is not understood at all. It certainly is different than what's seen in other respiratory viral infections, even severe ones. So evidence, this is a study that I just wanted to very briefly mention that appeared a little while ago, talking about inflammation in the CNS. And this is in this manuscript, Yang et al. Did new single nucleus transcriptomes from 8 COVID-19 and 14 control autopsies. Again, found no evidence of virus infection, but looking at choroid plexus cells, found there was upregulation of interferon and complement associated genes. And the authors also noted macrophage infiltration into the choroid plexus. So supporting the notion that there's some untoward inflammation going on mediated by factors we don't understand that's causing dysfunction. And in the cortex, there was increased numbers of T cells, again, without virus. Microglia showed upregulation of genes associated with disease, such as complement-associated genes, and downregulation of homeostatic markers, such as PTRY12. And astrocytes also showed increased expression of inflammation in astrogliosis genes, and with increased expression of chitinase, which is a neurotoxic protein. So all these things are pointing to some sort of inflammatory process causing disease in the brain, probably in other organs as well, but those are not as well studied. And so in terms of SARS-CoV-2 infections of the CNS, and I mentioned already the anosmia, but this is something that was intriguing to us. So I'm going to spend most of the rest of the time talking about this and then speculate on possible implications of what the anosmia means. So anosmia is a well-established complication of COVID-19. It generally resolves within a short period of time. However, it may persist for long periods of time with variable resolution. And it also can be associated with parosmia, which is misidentifying a smell, and phantosmia, where one smells something that's not there. And some of these aspects can be investigated using mice, and that's what my lab does. We mostly work with mice. So I'm going to describe some of our work with using mice. So the types of olfactory dysfunction that can occur after a viral infection can be several kinds. Some can be short and some can be long-term. The transitory or short-term dysfunction can include conductive or mechanical losses. So this is the obvious obstruction of the air of the nose or of the nasal cavity. Mechanical losses due to congestion. These result from blockage of inspired air due to local inflammation and edema of mucosal tissue in the olfactory cleft and upper nasal passages. This is what happens, of course, when people have colds. Then there can be sensorineural loss, which is the olfactory epithelium and cranial nerve 1 are infected. And this kind of dysfunction can be divided into two types. One of them is altered quantity or function of odorant binding receptor molecules and dysfunction of the neurons themselves, or they can be central dysfunction. And this can be confined to the olfactory bulbs, or it can be also isolated to upper order regions that are connected to the olfactory bulb, such as the piriform cortex and orbitofrontal cortex. And these are particularly important when one thinks about the implications of anosmia and possible long-term neurological sequelae. And the chronic or permanent dysfunction, we are seeing some of this now. There's possibly loss of olfactory epithelium, but this has to be because of the loss of neural stem cells, because the olfactory epithelium is regenerative. There can be disruption of the processing networks, and there can be, because of this, there could be uncertain functional recovery. And I'll mention that a little bit in the next slides. And we just published a review on this in Lancet Neurology with Zidakis as the first author. So this is one case that we saw at the University of Iowa. This is a patient who was a 46-year-old person who, at 4 days post-diagnosis of COVID-19, succumbed to the infection. And what we did is we were able to get nasal tissue, and what we saw was infection of sustentacular cells. That's what's shown here in the magenta color. Sustentacular cells, which I will spend a little time talking about, are the supporting cells for the olfactory epithelium. There's no evidence in this particular patient of infection of neurons. And this is, in fact, what's generally found across the many studies, is that neurons are not infected with COVID-19, but in the nasal cavity, you can find infection generally in sustentacular cells. And accompany these protein, here the virus is labeled in red, are these green cells, which are myeloid cells, which have infiltrated the areas near the infected cells, and it's the primary immune response. And again, this is a theme. I think we're going to see lots of evidence for myeloid cell infiltration into areas of virus, or as I'll show, maybe areas that don't have virus, but these inflammatory mediated changes seem to include myeloid cell infiltration. So this is one of the things that we first noticed. A student showed me this back in the beginning of the summer. And this was a study, not a study, this is a slide from Amazon. And this is about scented candles. And it's the proportion of reviews mentioning a lack of scent over 2020. So most of the time, there's a low level of people complaining that they received scented candles, but they couldn't smell them. But if one looks here, there's a big uptick in October and November. And this is, of course, when COVID-19 was taking off in the US in one of its spikes. And so this was a hint that anosmia was a big time problem in the United States. So then about the same time, we heard about this patient. This patient first in March 2020 was noted to have to have lost her sense of smell and taste. So she couldn't smell food, she couldn't smell perfume, and otherwise she was perfectly fine. And we know that she was exposed to COVID-19. It was March of 2020. She was at a wedding, not wearing a mask, and her daughter had recently returned from California, where there was a major outbreak of COVID-19. And so at the time, we didn't think much of it. She didn't think much of it, though within a few days, anosmia and ageusia were mentioned as possible manifestations of COVID-19. Anyway, after about 8 months in November, she mostly had recovered her sense of smell, but by 8 months, she had not completely recovered. And this was actually useful for her financially because she really liked fine wines. And now she could no longer tell the difference between a fine wine and a not so fine wine. So she ended up buying less expensive wines. So not a good trade-off, but a small trade-off. And then she became interested in November 2020 about whether her anosmia was in fact caused by COVID-19. So we did testing for her and we found out that she had an antibody titer of 1 to 48, and she had a T cell response shown here in these dots here in cells stimulated by the N protein or the spike protein. And so we concluded that she had COVID-19 back in March. And she's still not completely better. So partly for these reasons, we began to investigate anosmia in mice. And so mice are not infectable directly with SARS-CoV-2, at least not with the original strain of SARS-CoV-2. And to do so, to obtain infection, there needs to be 1 to 2 amino acid changes in either mouse ACE2 or the virus. And the way one can do this, one can either modify the mice so that it expresses human ACE2. This is either done by a transduction with a adenovirus vector, similar to the ones that are used for the vaccine, or the generation of ACE2 transgenic mice, or generation of mice in which the mouse ACE2 is replaced by human ACE2. In all cases, making it so that the natural virus, the original virus, can infect mice. One can also modify the virus so it can bind to mouse ACE2. Well, it turns out all these things came together naturally because the key residue that's important for making the virus infect mice is one called N501Y, a change from asparagine to a tyrosine of position 501. And it turns out that's present in many of the variants of concern, such as the beta variant from South Africa and the P1 or gamma variant from Brazil. So many variants are able to bind mice ACE2 directly now. So some of these changes are no longer needed. So we did another approach. We first used these K18 transgenic mice. These are mice that we had generated for the SARS epidemic in 2003 or so, 2005. And these mice were very exquisitely sensitive to SARS CoV and not really useful because the brains became infected. However, with SARS CoV-2, they're much less likely to develop a brain infection, particularly with the original strains of SARS CoV-2. And so therefore they're useful for studies. Sometimes we see brain infection, and this is much less if mice are older. But this is an example of a mouse that did have a brain infection. There's overwhelming infection of the brain. That's what the brown staining shows for the viral antigen. But also more interesting are these thrombi, which we think are important in the human disease and which we see occasionally throughout these mice. And because we were interested in anosmia, we then focused on the nasal cavity and the sinus. And what one can see here is that there's extensive infection of both the sinus, that's shown here with the brown labeling around the inside of the sinus, and also in the olfactory and respiratory epithelium. That's what the brown staining is here. And one can see disruption. This is day 5. You can see some destruction of the nasal epithelium and olfactory epithelium. More interestingly are shown the results shown in M, N, and O. Here what we see is very clear infection of these long cells. These cells are the sustentacular cells, which I mentioned previously, and which are important for supporting neurons, but are not neurons. And one can see very nicely the full extensive infection of these cells in these 3 panels. And this is another illustration showing that sustentacular cells, which are the red lines here, are infected with the viral protein, but OMP, which is the marker for the olfactory neurons, is not at all coincident with the cells that are infected with virus. So one can see here these nice olfactory neurons and then these infected sustentacular cells next to them. So now the question is, and we think this happens in people as well, as I showed earlier. So the question is, why is there anosmia if only the sustentacular cells are infected? And this is certainly something that we have some ideas about, and I'll show a little bit of data, but we don't really know. But I think it's really key to understanding this process, because this goes along with the idea that neurons themselves are not necessarily directly infected, but there are certainly effects on the neurons. So to do anosmia, we, and in this case, it being Abhishek Verma, a postdoc in the lab, we did several tests. So I'm not going to go through all of them, but one of them we did was to use male and female mice in a scent discrimination test. And in this case, we used a female mouse, and we exposed her a novel scent and a familiar scent. And mice like to explore. So normally mice would spend all their time around the novel scent, and that's what you're seeing here in the blue bar. This is the number of seconds that mice spend on the novel scent as compared to the familiar scent. And you can see there's a big difference here. But by 2 and 3 days after infection, there was no difference. The female mice spent as much time with the novel scent as with the familiar scent. And then Abhishek also did experiments with finding food. In this case, if you give a mouse a cookie, one of our favorite childhood books. And what we found is that in this case, if you look at male mice, this is how long an uninfected mouse takes to find food. And this is how long an infected mouse takes to find food. And what one can see is that by 3 days post-infection, mice take longer to find the food, and many of them cannot find the food by the end of the experiment. And we see the same results with female mice, if not more striking. Here again, many of the mice at 2 and 3 days post-infection could not find the food. On this side, I just show one thing that I won't spend much time on, but we treated mice with convalescent plasma, which prevented death and minimized lung infection. And still, these mice still have anosmia. So even though mice are mostly protected from the infection, they still develop anosmia because the nasal cavity is not protected from infection. So then we also did the same sort of experiments with our mouse adapted virus, which I haven't described yet in any detail. And this virus can infect normal mice, and because we added the N501Y, and then we also passaged it through mice to make the virus a little more virulent. And when we used these mice to look for anosmia, what we can see is that, again, at day 2, there's not much change. They can still detect food. They can find the food in the buried food test. By days 3, 8, and 14, they had a lot of trouble finding food, but by day 21, they were mostly recovered. And the same thing with the scent discrimination test. They spent most of their time around the novel scent in this case. And by day 21, that was true as well. But in between, these female mice had trouble telling novel from familiar scents. And then if one looks in the epithelium, it's mostly recovered by day 21. There's a little dilation of the Bowman glands, according to our pathologist, which I haven't marked and it's hard to see. So what's causing all this? So one of the things we first observed was that there was extensive loss of cilia, and that's shown here in these panels. In an uninfected mouse, green is the cilia, and you can see nice labeling across the whole epithelium. But after 6 days of infection, much of that labeling is disrupted so that there's only cilia in certain places. This occurs even though only a limited number of sustentacular cells are infected. And then if one looks for viral RNA, one can see extensive viral RNA, as I showed previously, at day 4 after infection. And then by day 6, it's resolved. Day 9, there's very little left. And by day 15, there's very little left. There's almost none left. So viral RNA is disappearing a little more quickly than the anosmia is resolving. This just shows the amount of viral RNA still left. At 3 days post-infection, this is by qRT-PCR, and one can see very high levels at day 3 and 6, but this is on a log scale and almost nothing by day 12 or 30 post-infection. And so one of the striking things we observed, we asked whether our olfactory receptors downregulated, could they be explaining part of the problem? And these results, I think, are pretty remarkable. This shows expression of 10 different olfactory receptors. And in an uninfected mouth, there's a huge variability in expression, but they're still all quite high. But by the time one looks at 2 or 6 days after infection, in almost all cases, expression of these olfactory receptors is almost at baseline. And then by 12 days, it starts increasing again. So showing that olfactory receptor downregulation may be contributing to the anosmia we see, at least to the acute anosmia. And then what's going on in terms of cellular infiltrates? Well, it turns out that there's not really much change in the cellular infiltration in total. I'm not going to show those data. And there's not much change in cytokine or chemokine expression, which surprised us. But this is the change that we found that was most noticeable. This is a stain for myeloid cell infiltration, and the numbers of myeloid cells doesn't change very much with infection. But what one can see is generally the myeloid cells are in the basal area. These are the green cells here labeled with IBA1, a marker for myeloid cells. And after 4 days of infection, these cells have migrated to be next to these infected sustentacular cells and throughout the olfactory epithelium. So how this is actually working is something we don't understand, but it seems as if these myeloid cells are infiltrating and there's widespread destruction of cilia, which is where the olfactory receptors are located. So this is all occurring as some consequence of sustentacular cell infection. And this is another experiment that's related to this, but a little different. So this is asking whether these cells now can be found in the brain. And here we're not looking specifically at olfactory connections in the brain, but just rather in the brain in total. And what we've used is we've infected mice with using our mouse adapted virus, and we've infected special mice. These are mice that express the D-tomato, the red stain, only in cells that are of the granulocyte monocyte precursor. So only these cells are labeled. We're not using any immunostaining. And what one can see here is that in mice after infection, there's a few of these cells in the uninfected brain, but in a young mouse after infection, there's lots of them throughout the brain. And in middle-aged mice, there may even be more. But again, they're found quite extensively found in the brain. We haven't looked yet at later days after infection, which is clearly the key thing to look at, because we want to know if these mice have any of the signs of long COVID-19 or anything that'll help us understand long-term sequelae. And I should say these mice have no evidence of virus infection. So for unknown reasons, these myeloid cells have migrated into the brain. And we don't know why, but we think this may be helping us understand some of the sequelae. And I didn't mention it because we're just getting preliminary data, but even our mice, even after a long time after recovery, their sense of smell is not completely normal. So we think we may have a system here for possibly studying some of the long-term effects of anosmia. So the summary of these anosmia experiments is that mice infected with SARS-CoV-2 are useful for studying anosmia. Female mice develop more severe anosmia than males. And in patients, COVID-19 is more severe in men, but there's different reports differ on whether males or women or men have predominant more anosmia. The original report said females had more anosmia, even though males had more severe disease. But I've come across recent reports that say, no, males have more anosmia. So I think this may still be something we need to firm down, make firmer. And the mechanism of action of anosmia is not really clear. The neurons are not infected, but the sustentacular cells are. So we're wondering if this has something to do with olfactory sensory neuron homeostasis or these inflammatory mediators, which we cannot identify whether they're somehow indirectly affecting neuronal function. And then finally, this is just something I get from the review that I mentioned early on by Zidakis et al. And this is something we thought about in the review. Could this olfactory system dysfunction have anything to do with long-term COVID-19 sequelae? And what this says, without reading the whole paragraph, is that we don't really know why by COVID-19 long haulers occur. But one possibility is that people who recover from COVID-19, this chronic recrudescent or permanent olfactory deficit may be prognostic for increased problems of other parts of the brain and spinal cord, which themselves result in either neurological sequelae or neurodegenerative disease in the long term. We, of course, don't have this information yet. And so the idea would be that an inflammatory stimulus from the nasal olfactory epithelium to the olfactory bulbs and primary, secondary, and tertiary connections of the bulb might accelerate some of these processes and result in neurodegenerative disease. And then there could be persistent olfactory impairment and perhaps impairment in other parts of the brain. So this is something that we don't have evidence for, but we're concerned is a possibility. So with that, I just want to acknowledge the people who did the work. Abhishek Verma did the anosmia work, and Roy Wang and Jian Zhang did most of the animal work. And then I wanted to thank my long-term collaborator, Paul McRae, and Kun Lee in his lab. And with that, I will stop and take any questions.
I'd like to thank